Hi, this is Pavitra Lama and this is my slide presentation for proteome analysis in Escherichia coli sample. Let's start with introduction. Bacteria keep on constantly monitoring changes and adjusting at molecular levels throughout the various stages of growth. Escherichia coli is an excellent model in biotechnology for examining proteome analysis, including its expression and steps under various physiological conditions for adaptation in mammalian gut. The methods such as 1D page, 2D page, and mass spectrometry are the techniques which have been used previously for proteomic research. STS page is a significant method for the separation and evaluation of molecular weight of the protein, whereas mass spectrometry is an analytical technique that measures the mass to charge ratio of ions. In our study, we plan to work with some of this methodology for proteomic analysis in treatment type of E. coli against a control type of E. coli. Here are the steps for proteome analysis. The first one is extraction of DNA. It is followed by determination of protein concentration, protein fractionation, angel trypsin digestion, and then second dimension STS page followed by IEF, that is isoelectric focusing, and then mass spectrometry and bioinformatics. Here are the software we used for data analysis. They are Quantity 1, Mascot, Pix database, Panther database. I want to talk about Quantity 1 software. Here are the advantages of using this software. It allows automatic lane and band detection, rapid molecular weight determination with choice of multiple regression model. It allows background subtraction correction of gradient gels. Quick and accurate concentration analysis is possible using sophisticated volume tools, and it is also a purity analyzer. Mascot is another software search engine that identifies proteins from peptide sequence databases using mass spectrometry data. Here are the advantages of using this software. It identifies proteins from an uploaded peak list using a technique known as peptide mass fingerprinting. It also combines peptide mass data with amino acid sequence and composition information usually obtained from MS tandem mass spectrometry data. It helps to identify fragment ions from uninterpreted MS data of one or more peptides. Let's move with Pix Studio. It is an excellent software platform with complete solution for discovery proteomics, including peptide and protein identification, de novo sequencing, database search, post-translational modification search with 500 plus modification, sequence variant and mutation search. We have the following advantage using this software. It performs LC-MS data analysis and statistics according to the experimental design, helps determine relative protein abundance changes between two groups by statistical analysis tool. The given two Venn diagrams are the result of our experiment produced by Pix Studio. The first Venn diagram shows the peptide number between control and treatment E. coli samples, and the second one similarly shows protein number between control and treatment E. coli samples. Here are another results from Pix Studio. The first pictures shows PSM score distribution, whereas the second one shows the distribution of peptide feature detection. Let's talk about Panther pathway. It is an ontology-based pathway database coupled with data analysis tools. Proteins are classified as family and subfamily, biological process, molecular function, and pathways. Here are two pie charts. The first one is for control E. coli sample, classifying protein according to molecular function, and majority of the pie chart is occupied by catalytic activity. Let's talk about the second figure. It is for control E. coli sample for classifying the protein according to biological process, and the biological process depends maximum on metabolic process. Here is another figure for treatment E. coli sample, classifying the protein according to molecular function, and majority of the pie chart is dependent on structural molecule activity. Catalytic activity. Now the next figure is Panther classification system, classifying the protein according to the biological process for treatment E. coli sample, and majority of the pie chart is occupied by metabolic process. This means the biological process in treatment E. coli depends on metabolic process. Here is our conclusion. Nearly 1186 protein in E. coli control sample and 1095 protein in treatment E. coli sample were found. The total molecular function gene found was 1087 and 1008 in control and treatment E. coli samples respectively. Some of the genes in treatment sample were downregulated or lost in comparison with the control sample. Overall, the presented information provides the most relative, absolute, and detailed quantitative analysis of bacterial proteome under normal condition and treatment level. Thank you.